Hello dear student, myself Bhanu Solanki, Senior Faculty Sri Chaitanya, Botany Department. Today we are going to discuss about the exam oriented specific and fine areas of NCERT in chapter Microbes in Human Welfare. Beside microscopic plant and animals, microbes are the major component of the biological system. Do you remember how many kingdoms among the living beings contain microorganisms? The answer is given in the same page. Microbes are diverse protozoans, means protista, bacteria, monera, fungi, microscopic animal and plant viruses. The answer, 3 kingdoms, monera, protista and fungi. Microbes like bacteria and many fungi can be grown on a nutrient medium and they form colonies. See, Bacillus bacteria, cocci bacteria, Bacillus again, bacteriophage is shown here there. Given member is a adenovirus. Adenovirus causes respiratory infections. Labeling of the bacteriophage, head, collar, tail, tail plate, pins also called tail fibrils, colonies of bacteria and fungi. Here there in this chapter the contribution of the bacteria are given. Microbes for the household products. First is given curd. Curd formation is facilitated by lactic acid bacteria. One of the well-known example is lactobacillus. Lactobacillus is a kind of lactic acid bacteria. What they do? Lactic acid bacteria produces acid, lactic acid, that coagulate and partially digest the milk protein casein. So it means curd is what? Curd is a partly digested and coagulated casein containing milk. That is curd. To form a curd, always there is a need of a small inoculum or starter that having millions of lactic acid bacteria. This conversion from the milk to curd enhance its nutritional quality. How? By increasing level of vitamin B12. In addition, they are very beneficial to checking disease causing microbes in our alimentary canal. Dough, which is used for making of the dosa and idli, also get fermented. The puffed up appearance of the dough due to the production of CO2 gas. Can you tell which metabolic pathway is taking place here? Then you must remember that alcoholic fermentation. Where do these bacteria come for fermentation? Through the pot, air, etc. A number of traditional drink, dough of the bread having baker's yeast, Saccharomyces cerevisiae. Toddy is a traditional drink of some part of the southern and India. It is also prepared from a fermented sap obtained from the palm, Caryota urens. Microbes also used for the fermentation of the fish and soybean and bamboo shoots. You heard about the tofu, that is a soybean fermented milk. Cheese is one of the oldest household product or item obtained by the fermentation processes. The characteristic texture, flavor and taste of the cheese is because of the inoculum or the bacteria that is using for the fermentation process. For example, large holes into the Swiss cheese are due to a bacterium And the name of bacterium is a Propionobacterium shamani. Roquefort cheese, due to Penicillium roqueforti, ripened by the fungi Penicillium roqueforti, gives the particular flavor to it. Then industrial products. It includes beverages and antibiotics. Unlike household product, for the production of industrial product, there is a need of large sized containers, means fermenters. Microbes, for the production of beverages, that is the yeast Saccharomyces cerevisiae. Same microbe is using for the bread making and for the beverage making also. That is why yeast is also known as brewer's yeast. Do you recollect the metabolic reaction which result into the production of the ethanol by the yeast? Of course, again, alcoholic fermentation. Depending on the type of raw material and the type of processing, whether the distillation happen or not, different type of alcoholic drinks are obtained. So, development of the different type of alcoholic drink depends on two things. One is the raw material and second is the processing. So on the basis of this, there are two types of alcoholic beverages, distilled and non-distilled. If they are non-distilled, then the alcoholic concentration would never be beyond 13%. If they are distilled, then there would be more than 13%. Beer and wines both are non-distilled alcoholic beverages, while whiskey, rum, brandy, gin, vodka, they all are distilled alcoholic beverages. Antibiotics. One of the significant discoveries of the 20th century is the antibiotics discovery. Anti means against the life. Bio means life. Together means against the life. But it is against the life of microbe, not against the life of organism who is going to consume them. One very commonly used antibiotics is a penicillin. Penicillin was first antibiotic to be discovered. Alexander Fleming, which working on Staphylococcus, once observed that the mold growing in one of unwashed culture plate around which Staphylococci do not grow. He found that it is due to a chemical produced by the penicillium, means mold, and he named it penicillin after the mold's name, Penicillium notatum. However, its full potential of the penicillin antibiotic was established by the Ernst Chain and Howard Florey. So this antibiotic was extensively used into the treatment of the soldiers wounded during the Second World War. So Ernst Chain, Howard Florey and Alexander Fleming were awarded with the Nobel Prize in 1945 for this discovery. Antibiotics have greatly improved our capacity to treat deadly diseases such as the plague, whooping cough, diphtheria, leprosy, etc. Then some chemicals, enzymes and other bioreactive molecules also. Aspergillus niger, source of citric acid. Acetobacter aceti, source of acetic acid. Clostridium butylicum, source of butyric acid. And lactobacillus of the lactic acid. For example, for products. Saccharomyces cerevisiae used for the commercial production of the ethanol. Microbes are also used for the production of enzymes. Lipase used into the detergents and are harmful, removing the oiling stains in the laundry. Bottled juice are more clear and clarified because of the pectinase and proteases. Streptokinase, obtained from the Streptococcus and modified by the genetic engineering, is used as a clot buster to remove the blood clots, especially into the patients undergoing myocardial infarction leading to the heart attack. Means lipase for the detergents, pectinase and protease for the cleaning of the juices, streptokinase obtained from streptococcus as a clot buster. Cyclosporin A is an immunosuppressive agent, so used during organ transplantation. Fungus Trichoderma polysporum is the source from which cyclosporin A obtained. Statin, another bioreactive chemical, obtained from Monoscus purpureus, it is also a kind of a yeast, is commercially used for the lowering of the blood cholesterol. How? The enzymes which are forming to the blood cholesterol get inhibited by this statin. So by enzyme inhibition, by competitively inhibiting to the enzymes responsible for the synthesis of cholesterol, it is working. So cyclosporin and statin and streptokinase are very important. Microbes in sewage treatment. Treatment of the waste water is done by heterotrophic organisms which are naturally present into the sewage. It happens in two steps. Primary treatment, it involves physical removal of the particles, large and small sized particles, through the filtration and sedimentation. So what is the basis of primary treatment? Primary treatment is a physical removal of the wastage, and it is based on it is carried out by the filtration and sedimentation. These wastage are removed into the stages. Initially floating debris removed by sequential filtration, then soil and small pebbles are removed by the sedimentation. The sediment that obtained is known as primary sludge, and the supernatant is known as primary effluent. Now this primary effluent is passed on for the secondary treatment. The primary treatment is a physical treatment by filtration and sedimentation. It removes floating debris, small sized grit and soil and pebble particles. Secondary treatment is a biological treatment. Primary effluent passed into the large aeration tank where it is constantly agitated mechanically. Air is pumped, aerobic process. It allows vigorous growth of the useful aerobic bacteria and they convert into the flocs. What is the floc? Flocs are the masses of the bacteria associated with the fungal filament in a mesh-like structure. It means as the breakdown of organic impurity is going on, this organic waste convert into the microbial biomass. The formation of the floc is a result of breakdown of organic impurities. While growing, these microbes consume to the major part of organic matter of the primary effluent. Consequently, there is significantly the biochemical oxygen demand of the effluent is decreasing. BOD is what amount of the oxygen that would be consumed by the bacteria during the breakdown of organic matter present in 1 liter of water. So it means as organic impurities get oxidized, BOD value will decrease. So the BOD test measures the rate of uptake of oxygen by the microorganisms into the sample of the water. Indirectly, BOD is the measure of organic matter present into the water. More the organic impurity, more oxygen is required for acid oxidation, means its BOD would be higher. The greater BOD of the wastewater means more polluting potential. Once the BOD of sewage or the waste material is reduced significantly, this material is then passed into the settling tank, secondary settling tank, where flocs get sediment, and this sediment is known as activated sludge. Means activated sludge is the floc, sedimented flocs. A small part of this activated sludge pumped back into the aeration tank to act as an inoculum, and remaining part means excess activated sludge is pumped into the anaerobic sludge digesters, where anaerobically growing bacteria lead its breakdown, digest it and convert into the biogas. During this digestion, bacteria produce a mixture of the gases of methane, hydrogen sulfide, carbon dioxide. These gases called biogas, and it can be used as a source of energy. So during secondary treatment, first breakdown of organic impurities in aeration tank, followed by sedimentation in settling tank, and then anaerobic sludge digestion. These are the 3 steps of biological treatment. Here they mention that no any kind of wastage can be freely released in our rivers and water bodies. Ministry of the Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save our major rivers of our country. Under these plans, it is proposed that to build a large number of sewage treatment plants so that only treated sewage may be discharged into the rivers. Microbes in production of biogas. Biogas is a mixture of gases predominantly containing methane produced by microbial activity, which may be used as a fuel. The type of gases produced during biogas production depends upon the microbes and the organic substrate that they utilize. In the example cited in relation to the fermentation of the dough, cheese making and production of the beverages, the main gas was CO2. But during anaerobic breakdown of the sludge, this is the biogas, means methane and carbon dioxide. However, certain bacteria which grow anaerobically on cellulosic material produces large amount of the methane along with the carbon dioxide and H2. These bacteria are collectively known as methanogens. One of the such common bacterium is the methanobacterium. These bacteria are commonly found into the anaerobic sludge during the sewage treatment. These bacteria are also present into the rumen part of the cattle. A lot of cellulosic material present into the food of the cattle. It is also present into the rumen. In rumen, these bacteria help into the breakdown of the cellulose and play an important role into the nutrition of cattle. Do you think we human beings are able to digest cellulose? No. Why? Because we haven't cellulase activity. Dung excreta of the cattle, means dung, is commonly called gobar, is rich in such bacterium. So dung can be used in generation of the biogas. So it is commonly known as gobar gas also. This is the scheme of a biogas reactor at biogas plant. This is the cement or concrete tank. This is dung and water mixed together slurry pumped into the digester, where the anaerobic breakdown happen, methane and carbon dioxide arise. As the level of gases increase, the upper cover will also increase and the gas will pass on to the utilization, and the waste slurry, that is called sludge, is withdrawn from here and transfer into the fields as a manure. The technology of biogas production was developed in India mainly due to the efforts of Indian Agricultural Research Institute IARI and Khadi and Village Industrial Commission KVIC. Biocontrol agent. Biocontrol refer to the use of microbes to control disease and pest. In modern society, the problem have been tackled increasingly by the use of chemical, but we know that insecticide and pesticide chemicals are toxic and extremely harmful to the human being as well as for the air, water, soil, etc. So their use is not a good idea, is not an eco-friendly method. Biocontrol is relatively more safer. In agriculture, controlling to the pest relies on the natural predation rather than being used introducing chemicals. There is a belief in the organic farming that the biodiversity furthers health, means more the biodiversity, more balanced the system would be. Therefore, the organic farmer works to create a system where the insects that are sometimes called pests are not eradicated but instead are kept at the manageable level by a complex system of the checks and balances within the living and vibrant ecosystems. This is the holistic approach that seeks to develop an understanding of the webs and interaction between the organisms that constitute to the field's fauna and flora. Why? Because if we were aware about these interactions, then we can create a more balanced system. The use of biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. An important part of the biological farming approach is what? That the farmer should be familiar with the various life forms of the organisms inhabiting to this area, their predators as well as pests, and also their life cycle and pattern of the feeding and their habitat preferences also. If all this information we have, so we can create an ideal organic farming system. Some examples are given here there. Red and black marking ladybird beetle, useful to get rid of aphids, and dragonflies used to get rid of mosquitoes. Bacillus thuringiensis produces a toxin, and these toxins are nowadays available into the sachets in a dried spore form, which mixed into the water and spread over the vulnerable plant, such as the brassica and fruit trees, and it will kill in the gut of the larvae, these toxins release and the larvae get killed. The bacterium disease will kill to the caterpillars but leave the other insect unharmed. Because of the development of the method of genetic engineering, this gene responsible for this toxin production or cryprotein, we have introduced into the plant to produce the many genetically engineered crops. One of the famous crop is the BT cotton that we will explore into the next chapter. Some more examples are given here there. For the treatment of the plant diseases, Trichoderma fungus is used. Trichoderma species are free living fungi. They were commonly used into the root system. They are effective biocontrol agent of the several plant pathogens. Baculoviruses are pathogens that attack the insect or other arthropods. Polynucleohydrovirus is one of the most majoritarily or most commonly used baculovirus. Viruses are excellent candidates for the species specific and narrow spectrum insecticidal applications. Means they will kill and destroy to the specific group of insect, not to all. That means only target insect will die, rest other useful or beneficial insect will not get affected. Microbes as a biofertilizers. In our present day lifestyle, environmental pollution is the major cause. The use of chemical fertilizers to meet ever increasing demand of the agricultural produces has contributed significantly to the pollution. Organic farming is one of the way that reduce this pressure, means use of the biofertilizers and biopesticides. Biofertilizers are organisms that enrich to the nutrient quality of the soil. The main source of the biofertilizers are bacteria, fungi and cyanobacteria. Some examples are given here. Nodules in the root of legume plants formed by the symbiotic association of rhizobium bacteria that fix atmospheric nitrogen into the organic forms and enrich to the soil. Other bacteria that can fix atmospheric nitrogen in the free living form are Azospirillum and Azotobacter. Three examples are mentioned here: Rhizobium symbiotically, Azospirillum and Azotobacter free living. Among fungi, mycorrhiza यानी कि symbiotic association of the root with the fungus. Glomus is one of the genus which contribute maximally into the mycorrhiza formation. What are the advantages of it? Plant having such mycorrhizal associations of the glomus are benefited. They are resistant to the root and bone pathogens. They are more tolerant to the salinity and drought. Their overall growth get increased and the development also improve. These are the and phosphorus at absorption. Absorption of the phosphorus from the soil and pass on into the plant are the main contributions of the mycorrhiza, glomus mycorrhiza. Absorption of phosphorus, resistance to the root-borne pathogens, tolerance to the salinity and drought, overall improvement of the growth and development. Cyanobacteria are autotrophic microbes widely distributed into the aquatic and terrestrial environments, both aquatic as well as terrestrial, may of which can fix atmospheric nitrogen, Anabaena, Nostoc, Oscillatoria. In paddy fields, cyanobacteria serve as an important biofertilizers, especially Azolla fern that has the colonies of Anabaena. Currently in our country, a number of biofertilizers are available commercially in the market and farmers use these regularly in their fields to replenish the soil nutrient and to reduce our dependence on chemical fertilizers. So it is the microbes in human welfare. It is a theoretical chapter, but 1 to 2 questions every year without any exceptions comes from this chapter. So just read thoroughly NCERT each and every paragraph, especially those areas that I highlighted in this chapter. You would not be in any kind of a trouble while any kind of a question come in exam. That's the motive of this program. All the very best. Thank you very much.